Hello, my name is Brian O'Connor. I am the software architect and owner of Nimbus Informatics, and I'm going to walk you through the products that we offer for next-gen sequence analysis on the cloud. In our case, we use the Amazon cloud, but the Nimbus products are actually based on, and we support, the open source Sequare project, which is actually designed to run on both private and public clusters. So in this tutorial, I'm going to show you how to log into the Nimbus portal and give you basic information about how to set up studies, samples, experiments in preparation for uploading data and analyzing data on the cloud. In subsequent videos, I'll show you how to actually do the injection of sequence data into the cloud and also how to trigger workflows and get analysis results back. At the current time, what we specialize in is whole genome, whole exome, alignment and downstream analysis and annotation on the cloud. And we've also worked with clients on genome assembly and other very interesting next-gen applications as well. So if you feel that we could possibly help you in your project, let us know. We're interested in doing one-on-one consulting work and also offering these tools as a service on EC2 for anyone that's interested in signing up for an account. So at this point, what we're going to do is we're going to log on to portal.nimbusinformatics.com. The easiest way to get an account with us is just to approach us over help@nimbusinformatics.com, and we'll set you up with an account and work with you on the particular projects that you're interested in analyzing on Amazon's cloud. So in this case, we've been redirected to our current server, and I'm going to go ahead and just log in. This information would be provided to you by Nimbus Informatics, assuming you've set up an account with us. Okay, on first login, what you're going to see is on the left-hand side, there's a variety of functionality, a variety of features that you can use. We're going to concentrate on the first one, but the idea here is that you can define your studies. That's really the first step in getting data analyzed with us on the cloud. You define your study, your experiment, your samples, and then you upload and associate that with real data that then gets processed through our workflows. So in this case, you can show the studies that you have, you can create new ones, you can do upload of sequence, and you can do bulk download, which is very convenient for pulling back information once your analysis is complete. Under the analysis tab here, you see that you can show the analysis that's running, you can trigger new analysis, and in the future we will have user uploaded workflows. That's a capability that we're working on right now very closely with OICR in Toronto for developing basically a software developer's kit that will allow anyone using our system to trigger their own workflows using the Nimbus Informatics infrastructure and underlying Sequare infrastructure on EC2. This tab here for sequencer runs is a very, very lightweight LIM system. It's similar to other LIM systems out there, but it's much more focused on just tracking the runs of sequencing instruments. For example, you could use this as a lightweight LIM system. It's not necessarily a full-featured LIMS that will track experimental protocols, for example, but it will let you associate the runs of instruments with particular lanes and associate those back with particular samples, which is helpful for tracking your downstream analysis. And you can see an example here where I've just set up an 8-lane Illumina GA2 run. Finally, under the My Account information, you can go in and log in and change your user password, password hint, that sort of thing. And there's also the ability to invite other users to the system. Okay, so going back to my studies, under this study, what I'm going to do here is I'm going to create a new study just as a test. So in this case, I'm going to call it test. I'm just going to put in a little gibberish here because I don't want to spend a lot of time filling these out. But you'll notice here that the overall study describes the whole overall approach for your project. And so in this case, I'm going to choose whole genome sequencing, and then my center is going to be gibberish as well. But you, of course, want to put in your real center information. So this study is now basically the entity that holds all your experiments and all your samples that are all kind of grouped together in one logical unit. So I'm going to go ahead and add an experiment. The experiment lets you basically define on a platform basis. So for this, in this example, I'm going to say a GA2. It's a GA2 experiment. I'm going to fill in a little gibberish for these guys, but of course you'd want to put in your real information. And then I'm also going to say that the library design is one called foo. And most importantly, I'm going to fill in the information about the library strategy. So at the experiment level, we're saying the particular platform, but we're also saying what this was, what library strategy was used. In this case, I'm going to use whole exome. So I'm going to select whole exome sequencing. And then finally, the final very important piece of information is this little descriptor language actually lets you describe the way that the reads are laid out. So in this case, I'm saying forward 50, reverse 50, so it's a paired end run. Okay, so now all my samples that have been sequenced in this way using this technique on the GA2, 50 by 50 paired end run, I can associate all those samples with that particular experimental design. So in this case, I'm going to say test, and I'm just going to fill in a little gibberish here and here. And then I'm going to say that this sample is from Homo sapiens. And there's other information that you can include too, information about adapters and regions. This is more for your bookkeeping. It's less useful for our system since our analysis workflows are primarily concerned with data input file formats and less about the actual metadata. But setting up this metadata properly is really, really helpful for keeping track of what experiments, what samples were associated with what analysis events. And so now you can see here that we have a test study, we have an experiment, and we have a sample all brought together in a tree-like structure. And you can decorate the study with multiple experiments. You can have multiple samples per experiment, and so on and so forth. So that gives you an idea of what this portal application is about. In a subsequent video, I will show you how to actually do an upload of data and have it associate down with these samples. So literally uploading a FASTQ file. And I will also show you how to use this analysis box over here for triggering whole genome, whole exome alignment, variant calling with GATK or other tools, and variant annotation with Anavar and other annotation sources as well. So that will be the next video, and I will see you soon.